So today I'd like to introduce the new instrument from Luminex, the xMAP INTELLIFLEX. The presentation is entitled Rich History of Enabling Groundbreaking Research. For those who are not familiar with Luminex xMAP technology, it's basically a bead-based assay where you can run on protein basis capture sandwich, competitive assays, and direct or indirect serological assays for instance, identification of proteins, also patient stratifications, also protein-protein interaction. Also, what can be done with this bead-based technology is genomic applications like SNP assays, mRNA assays, and nucleic acid sequences. It's an open technology. It means that you can build your assay yourself, but there's also a large kit content on the market. Luminex is a tech company, and actually the next speaker will be of one of our partners showing some more kit applications. So in short, there are a number of benefits compared with traditional techniques. If you look at multiplex technology, as an example, I took some data from 2 papers published last year and this year. And as an example, at 21-plex in 80 samples, basically, if you look at, if you do that with classical ELISA, you need around 42 plates. If you do it with a competitor assay like a planar array, you can do it in 3 plates. And if you do it with Luminex technology, you can actually do it all in one go in 1 plate. So if you then compare Luminex technology versus single-plex, obviously you use less sample, less time and labor, less expenses because you only do 1 plate rather than 42. If you build bigger panels, you get more data, more insights in your research, and it's quite flexible. And also better reproducible compared with ELISA. Hence, you will improve your scientific output. If you compare it with other planar arrays, the big benefit is basically if it's a bead-based assay that it's a suspension in liquid. So the physics and the kinetics are even faster. And because it's bead-based, there's also a high surface to volume ratio, also increasing the sensitivity of your assays. So the Luminex INTELLIFLEX is basically the best year of 25 years of Luminex xMAP development. It's the 4th generation machine, which is just came on market basically this month. So if you look back at the history of the company, in 1995, it was founded. First machine was the Luminex 100, which was upgraded to the 200, still on market. Then a number of years later, the FlexMap 3D came on market. A year later, the MacMix, which is now only as IVD on the market. And basically 10 years ago, we launched our new system, which is called the INTELLIFLEX, which is completely renewed. So optics are improved, hardware is improved. It contains a fully integrated system. And this system is modular compared to all other systems. That means that it's easier also to handle in the field. It has a reduced footprint compared to our current portfolio, the Luminex 200 and FlexMap 3D. So basically it's 2 megapixels in size. It has modules like the optics module, fluidics module, and plate module. And the PC is also embedded with the machine with a touchscreen. That means that also service in the field is way easier. If for whatever reason something will break down, it's just swapping a module and not taking out single components. And also there's no additional computer decks to the side. So basically what you see, this machine is what you put on your bench. It doesn't take any more space. The machine has multiple USB ports, also one on the side of the touch screen, and it has a network connector on the back, so you can put it in your local network. So compared to all other systems, it is quite alike, with the only exception that we now also have one additional violet laser. So basically, if you look at Luminex technology, normally the red laser uses to identify the beads. So the red laser hits the beads, and then the light is scattered to the side, and then you have 3 APDs basically looking at the 3 colors inside the bead, defining it which bead it is, between 1 and 500. And then the double discriminator or the red side scatter, which is basically defining it if it's a single bead or not. And if it's not a single bead, it will end up in an abort rate. The green laser will give the classical signal. Most of the time it's generated from PE. So that's the amount of target which is bound to your bead, which is then comes into the orange signal, the RP1. And then on the INTELLIFLEX, there's a new laser, which is the violet laser, a 405 nanometer violet laser, which comes into the blue PMTA chip, and that contains the violet and the second reporter. So if you would look how a classical assay with Luminex xMAP technology actually works, you see it here. So if you want to do, in this case, a free versus a bound drug assay, you coat your first bead with a specific antibody which will coat or capture your free drug or your free and your bound drug basically together. And then you come back with a specific antibody only detecting the free drug. Then the other bead containing the same coating antibody, you come back with the other classical, the bound target as a coating. And then you run these two as 2 assays through Luminex equipment. If you now would switch to an INTELLIFLEX, what you can actually do is take the 2 specific antibodies and actually label one with the classical PE and label one with a violet dye, and then run it as a target from the same bead. Hence, you end up with a dual reporter assay. And there are multiple violet dyes on the market. So why did Luminex choose violet? Here you can see the spectral overlines, and what you will see actually is that we chose a 4 or 5 nanometer laser, which is in the visible spectrum. So there's no additional regulations just for a classical laser. The violet dye has no spectral overlap of any of the other, the green and the red laser already in the machine. And because it's a violet dye, the wavelength is very short, and so it is an intense wavelength. That means that also there are intense reported dyes available on market, giving good signals in the machine. And what I already stated, there are multiple dyes on market, making reagents available. And then I will show you later on that there's no energy transfer between the violet and the PE channel. So there's more improved within the technology. Something what we call optical noise is basically taken out of the machine. And to assess optical noise, you can use Spherotech rainbow calibration particles. And what this basically is, you have these color-stained particles, which will give a fluorescence pattern. And here you can see on the upper figure, basically that if you look at the FlexMap 3D, that you can see the light green is still distinguished, and then the darker green and the yellow and the red, those peaks are basically moving in each other. And this is due to the optical noise from the electronics within the machine. Now the INTELLIFLEX is designed 10 years later, and of course technology doesn't stand still. And here you can see is actually that the INTELLIFLEX, the optical noise is taken out of the machine, and those 3 last peaks are separated in a good way, meaning that the optical noise is out. This means that also you can run high sensitivity assays with the machine. With one exception, is that if your reagents do not allow improved sensitivity, you will not get it out of the machine. So the hardware is now capable, but it still depends on the assay. There are also multiple detection modes in the INTELLIFLEX, and I will show you some data later on. But basically, it is fully backward compatible with all previous assays, and it will also have compatible measuring modes. That means that if you are already a Luminex user and you have an LX200 or a MacPix in your lab, you can actually choose that setting in the INTELLIFLEX. And if you would measure 10,000 MFI in your LX200 assay, you will measure 10,000 MFI in the INTELLIFLEX if you choose that setting. Same encounters for FM3D. If you run 160,000 MFI on your FM3D, you choose the FM3D setting in the INTELLIFLEX, it will also give you 160,000 MFI. And then there are two additional settings, basically the dual reporter setting, showing that you can measure 2 signals from 1 beat. And there's an additional high sensitivity mode on the machine, where the PE laser is boosted from 15 milliwatt to 50 milliwatts, meaning that the intensity of the laser is improved, and that will also give you an extremely high MFI. That means that if you normally would run like 15,000 MFI, if you then run it on a 50 milliwatt output, that will result in an MFI of several million. So basically, there you can actually give an additional boost to your signal if your assay will need that. So that's an additional feature of the dual reporter machine. The INTELLIFLEX will come on market as 2 versions. One is the basic model, which is the xMAP INTELLIFLEX. So it will have a capacity like our FlexMap 3D, 500 different beats, dynamic range 5.5 log, slightly bigger than the FM3D, and then compatible 96.380 format plate. And already stated, PC and monitor are fully integrated in the machine. Reading times around 20 minutes for a 96 well plate and 75 minutes for a 380 well format plate. As showed, it's fully modular. Also on front there's a sliding door, which means that if you want to clean out your probe or need to replace the probe, it's quite easy. And then the machine will also come as a feature which is called the INTELLIFLEX DRSE, which stands for Dual Reporter Side Eject. What it actually means is that is a machine where the additional laser, so the violet laser, is in, and it will come with a feature that you also can unload your plate or load your plate to the side of the machine and not on the front. That means that the machine is extremely automated friendly, because if you now place your machine next to a robotic system, it means you don't have to place your system in the deck, and that will save you a lot of deck space. So let's talk about backward compatibility testing and some dual reporter applications. And then the next speaker will actually show a lot more data, not on the dual reporter, but on the kits for oncology. But this is data I got from the next speaker, where they run some backward compatible assays, and you can see here they run it on several machines, in this case the Luminex 200 and FlexMap, and then they run it in several settings on INTELLIFLEX, what we already discussed, like the LX200 low PMT or FlexMap 3D high PMT in purple. Take-home message of this one, and this was actually from a 45-plex, that if you run biological samples in any comparable mode in any Luminex machine, you will find the same biological data. So if you buy a Thermo kit, it doesn't really matter if you run it through the LX200, a FlexMap 3D, or an INTELLIFLEX, you will get your same biological data. So some dual reporter assay, and I've just picked a serological assay in this case for COVID, not an oncology type assay, but this is just for an example because we just had some data on it, as the machine is just out for a month. So basically what you will do in this case, you will code a target on the surface of the bead, then you incubate with your sample. And if you do a serological assay, it's not only IgG which is bound to your target, it could also be IgA, IgM, D and E. You incubate with your sample, you wash it, and then you come back. And then you can come back with your classical PE, like in this case an anti-IgG, and a violet dye, an anti-IgA or an anti-IgM. And then you run through the INTELLIFLEX, where the red laser, as stated, will detect the beads, and then it goes to the green laser for the PE signal and the purple laser for the violet signal. Now, of course, we want to see that if you run either the PE signal or only the violet signal, that there's no spillover in energy or light emitted between those targets. So this was a proof of concept assay where, and I'll just talk you through it, in figure A, there was only a PE antibody present in the sample. In figure B, same sample, there was only an IgM, super bright antibody present. And then figure C is that it was done as a dual reporter. And what you basically see, and that's the take-home message of this slide, that if you do a dual reporter assay, that the curves are exactly overlining if you run it either as a single or as a dual reporter. That means that these signals are not interfering with each other also in a biological assay. This is also from the COVID kit from Luminex. Basically, this was a study where we looked in various individuals which were COVID naive, haven't seen the virus yet, and were vaccinated with BioNTech Pfizer, and it was a kinetic study. And then we looked at antibody responses in time. And basically what you can see after 9 to 10 days after the first shot, you see a massive production in this case in IgG and IgA. It also encounters of IgM, which is not depicted in this figure. And then on day 24, there was a booster given, and again you can see that as an antibody response as PE and IgE, PE, and IgA. The middle picture for those who are doing COVID research probably know it, but this is basically plotted N versus RBD, and where you can actually see is that in blue that were biological samples pre-COVID, so they haven't seen the virus and you don't see any antibody response to either N or RBD. And there, here you can distinguish those who had a natural infection, which are also N positive, which are in red, and those who only are vaccinated, which are in black, they are N negative. Now, what we also did, we run a sample set of 180 biological samples, which we run we're using IgE PE labeled on MacPix, LX200 and FN3D. And then what we next did is we took the same antibody clone and then we run that as an IgA, as a brilliant violet 421 on the INTELLIFLEX. And here you can actually see the correlations between the targets, which are again matching quite well, showing that the INTELLIFLEX is compatible. It is backward compatible, and it doesn't really matter if you run it either as PE or in the violet channel. You will get the same biological data. Well, what kind of dyes are on the market? And this is just a few we tested, in this case for proteomic assays. So Super Bright is on the market, and Horizon Brilliant Violet 421 is on the market. And the nice thing is that these two molecules are available as streptavidin. So if you are having a classical biotilated antibody, you can use these two markers to do a dual reporter assay. And then the nice thing is there are tremendous amount of directly PE labeled antibodies. So this gives you a free choice making a dual reporter. What you also can see is Alexa 405 is quite dim, as is Dialight 405, and we also test Horizon 450, but that's basically out of the range of the measurements, and that you also can see it only gives you 5% of the peak MFI Super Bright. Alexa 405 is quite dim, however, very useful, for instance, in nucleic acid, because there it is thermostable, so it's easier to use for molecular assays rather than for protein assays. Now, what kind of applications can you think about it? You can do like phosphorylation, so you can do a total target, a phosphorylated target. You can do protein-protein interaction. As discussed, antibody isotype like IgM, IgA, or class switches between IgG1 and IgG3. Also neutralization assays. You can do free versus bound drug, receptor ligand interactions. Also, as treatment in oncology, you can, for instance, do selection on bispecific antibodies or fusion proteins to see if your targets are on there. So basically, the sky is the limit. So as a summary, I believe that the INTELLIFLEX is basically the best of 25 years of xMAP experiments. The machine has a number of hardware upgrades, especially the reduction of the optical noise means that the machine is capable to run on a more sensitive way. Side ejection might be very useful for automated streets. If you get a dual reporter, 500 bead sets, you can harvest actually 1,000 data points per target. And then as an additional feature, you have the high sensitive option on the PE label. And then it measures a bigger range than our other equipment. And of course, then the dual reporter capability. Hence, there's some other features. The software is renewed. It's completely different. It looks alike, but it has different backbone. There are more export versions. It's very easy to use, both the hardware and the software. And for instance, also some upgrades, like there's a barcode scanner in the machine, so you don't have to type in or load anything from CD. It's just scanning a barcode, and then all your column variables are in. And as stated, it is fully backward compatible with our current portfolio. So with that, I'd like to conclude my presentation and give in the word to the next speaker.
Okay, thank you. So in the second part of this webinar, I would like to provide insights into assay solutions and applications based on the Luminex multi-analyte profiling technology to support immuno-oncology research. So to further elucidate the interplay of tumor cells and the tumor microenvironment and better understand the mechanisms that underlie cancer also in view of potential therapies, exploring all facets of the immune system and immune checkpoint pathways is key. So the figure on the left underlines the complexity of the immune system, which is a network that involves a multitude of cells and molecules, including also a large number of soluble factors such as cytokines and chemokines that shape local and systemic immune responses, which overall depend on the critical balance between stimulatory and inhibitory mediators. So there's a dual role of inflammation also in the context of cancer that involves several cytokines which are either cancer promoting or rather support anti-cancer responses together with soluble receptors and ligands of T cells and natural killer cells, as summarized in the figure on the right, that are critical players in immuno-oncology, which rapidly evolves and emerges as a research field focusing on innovative therapeutic approaches that take advantage of the ability and an individual's immune system to fight cancer. So among the key research areas, as shown here in immuno-oncology, cancer vaccines have evolved a strategy based on antigens and neoantigens that are released by cancer cells upon cell death to stimulate a strong immune response, besides the chimeric antigen receptor T cell or CAR T cell therapy that further expands the adoptive cell therapy, relying on the transfer and reinfusion of a subject T cells after specific genetic modification, which finally enables them to recognize and destroy cancer cells. And the immune checkpoint inhibition as another approach that is based on therapeutic antibodies targeting immune checkpoint pathways as a way to attack tumor cells. The immune checkpoint pathways are critical in balancing immune responses in order to protect tissues from damage when the immune system is active, but can also be dysregulated by cancer cells that use them as a mechanism to ultimately evade immune surveillance. So as outlined here on this slide, among first immunotherapies to market were targeted to blocking effects of cytotoxic T lymphocyte antigen 4, CTLA4, and programmed cell death 1, PD1, with several antibodies actually approved as immunotherapeutic drugs. CTLA4 is a negative regulator of T cell activation and proliferation, and upon binding to its ligand, plays a role also during priming of these T cells. Blockade of the CTLA4 receptor by specific antibodies, however, can enhance T cell activation and thereby drive anti-cancer immune responses. The immune checkpoint protein PD1, on the other hand, is a negative regulator of apoptosis, and by binding to its ligand PD-L1 that is expressed often by a cancer cell, it can mask cancer cells from T cell recognition and finally also results in attenuated immune responses. So since the discovery of cancer therapy based on inhibition of negative immune regulation, such as via CTLA4 and PD1, A lot of strategies and agents have been developed to target these immune checkpoint markers, which are actually in clinical trials and also used in combinatorial therapies. And investigation of immune checkpoint pathways still continues in order to identify new potential drug targets and predictive biomarkers for checkpoint immunotherapy. That is not only important to optimize potential therapeutic benefits, but also to minimize possible side effects and guide combination therapies. So to support cancer biomarker discovery and verification across these key immuno-oncology research areas, we actually provide two different applications and InvitroGen assays for highly multiplex protein and gene expression studies on one instrument platform, so they are compatible with any of the Luminex instruments as previously presented. On the one hand, we have the hybridization-based Quanti Gene Plex assays with incorporated branch DNA technology that allow for accurate RNA quantitation of up to 80 targets per well directly from a blood, lysed or tissue homogeneous sample without the need for RNA purification. On the other hand, ProcartaPlex immunoassays enable scalable and reproducible protein quantitation with the simultaneous detection of actually up to 65 targets in a single sample of plasma, serum, and other bodily fluids that require only 25 to 50 microliter of sample volume in the 96-well format, and even less in the 384-well format that is now also available as a unique format of pre-dispensed lyophilized beads on a plate next to a liquid formulation with a bead premix provided in a vial. So most importantly, these multiplex assay platforms can also be customized by selecting any RNA transcript or by choosing from our broad list of protein targets for human, mouse, and other species that cover chemokines, cytokines, but also relevant biomarkers for the immuno-oncology research space and many other areas of biology. So to better understand also the molecular basis that may underlie cancer-related immune responses, the Human Immuno-oncology Checkpoint Panel on the Quanti Gene platform enables the simultaneous measurement of 50 checkpoint modulators and immune cell type markers, as listed here in a 96 or 384 well format. Besides, the custom research panels can also be designed incorporating particular targets of interest and allowing the relative gene expression data to be obtained for these selected targets from the same sample. As shown here as an example, in a publication in the Journal of Immunotherapy of Cancer, a custom Quanti Gene Plex panel was used for gene expression profile analysis in the preclinical discovery and development of a novel monoclonal antibody that binds to the PD-L1 checkpoint ligand. And to evaluate the effects of this antibody, a high-content molecular analysis were performed on cell lysates from mixed leukocyte reactions, as depicted by the volcano plot in the upper left, as well as on tumor tissues collected from several xenograft mouse tumor models, as illustrated by the plot on the upper right. So analysis of in both models revealed finally a prominent T-cell response upon addition of this blocking antibody, consistent with T-cell activation and also involvement of the interferon gamma pathway. Furthermore, by comparing a single agent treatment versus a combination of PD-L1 and CTLA-4 antibody treatment, as shown in the Venn diagram below, gene expression changes indicative of immune activation was observed for single anti-PD-L1 treatment, as well as enhanced T-cell activation in response to single anti-CTLA-4 treatment. And furthermore, a combination resulted in further upregulated Th1 type responses. So overall, this highlights how Quanti Gene Plex assays can effectively support the evaluation of potential novel immunotherapies, as demonstrated by the ability of this antibody to effectively block the PD-L1, PD-1 axis to drive a more effective anti-tumor T-cell immunity. On the ProcartaPlex platform, in recent years, a main focus of development activities was also on immuno-oncology biomarkers for investigating immune checkpoint pathways, functionality in effector T cells and natural killer cells. So as schematically shown in the 7-step process of this cancer immunity cycle in the figure on the left that summarizes how the immune system combats cancer cells, initiated by the release of cancer cell antigens upon cell death, followed by the uptake and subsequent presentation of cancer antigens, T cell activation and trafficking of activated T cells to the tumor site with infiltration into the tumor, and finally the recognition and killing of cancer cells associated with the release of antigens that further drives the progression of this cycle. So each of these 7 steps is tightly regulated, involving a multitude of checkpoint molecules, other cell surface receptors, but also soluble factors such as cytokines and chemokines. And for many and almost all of these factors, we provide multiplex panels, such as for pro and anti-inflammatory cytokines that regulate step 2. We provide a complete portfolio of chemokines and adhesion molecules that regulate T cell trafficking and infiltration. And finally, our immuno-oncology checkpoint biomarker panels particularly focus on steps 3 and 7. T cell activation and killing of the cancer cells, including most important co-stimulatory and co-inhibitory molecules that are often engaged by cancer cells to alter these activation pathways and are also targeted by immune checkpoint inhibitors. In addition to this important role of the adaptive immunity, similar processes and checkpoints can also be found during innate immune cell activation, as depicted by the figure on the right, showing a lot of receptor-ligand interactions of natural killer cells that are also to be explored as potential therapeutic targets that might mediate anti-cancer immune responses. And the figure furthermore points out that immune checkpoint molecules are primarily considered as receptor-ligand pairs, but a lot of soluble isoforms and shed variants are described for the majority of these targets that can function as immune adjuvants or decoy receptors, thereby regulating the checkpoint pathways, but which might also impact the clinical efficacy of individual checkpoint modulator drugs. So as a consequence, the immuno-oncology checkpoint biomarker ProcartaPlex panels have been specifically designed and optimized for measurement of key immuno-oncology analytes in human or mouse serum, plasma, and cell culture supernatant samples. And this is illustrated by this bar chart. So in a screen of more than 70 human melanoma samples finally confirmed that all these soluble forms of checkpoint molecules that are included in the immuno-oncology checkpoint panel 1 and 2, which each include 14 targets, are indeed detectable in serum. Further evaluation of the ProcartaPlex Immuno-oncology checkpoint panel 1 showed that the expression patterns of these soluble markers are dependent on disease state and cancer treatment. So as demonstrated by the 2 graphs, reduced levels of PD-1 were detected in melanoma samples comparing early treatment to pre-treatment, showing characteristic responder profiles which might help to pre-select and stratify subjects who may benefit from a certain treatment, and further highlights that analyzing these soluble checkpoint molecules will provide valuable information for early evaluation of treatment responses. Moreover, levels of soluble checkpoint markers could also be determined in human breast cancer samples, as shown here, before and after chemotherapy, using the immuno-oncology checkpoint panels as previously published. And there is evidence that chemotherapy-induced tumor cell death is related with the release of intracellular molecules, such as the cell death marker HMGB1 into the microenvironment. So as demonstrated by the box plots, soluble CD27, soluble CD80, and soluble PDL2 correlated with HMGB1 levels and were increased in human samples with a better response to chemotherapy and a better 5-year survival. And these findings suggest that the immune status differs between donors with increased HMGB1 levels in response to chemotherapy as compared to donors without increase, and further underlines the relevance of immune checkpoint marker analysis to better adapt treatment strategies and responses. So apart from the human pre-configured panels, the mouse ProcartaPlex Immuno-oncology checkpoint panel 1 and 2 allow also analysis of specific soluble receptors and ligands to support studies on the response to potential therapeutic treatment in mouse models, and have been evaluated by a collaborator here in tissues from mice. So as demonstrated in the two bar charts, in total analysis of 11 analytes in tumor tissue extracts from treated and non-treated mice revealed a distinct protein profile with differentially expressed proteins in the treated as compared to the control group, suggesting a role for these proteins in tumor progression. So in addition to the immuno-oncology checkpoint biomarker panels, it is also of great interest to analyze a broader set of immunoregulatory soluble factors that provide significant insights and a more comprehensive profile and picture of an individual's immune status, such as the human immune monitoring ProcartaPlex panel that is actually our largest available panel on this multiplex platform and enables efficient analysis of immune response profile, biomarker discovery and verification by combining 65 cytokine, chemokine, and growth factor targets in a single well. Data analysis is furthermore enhanced also by an intuitive, easy-to-use software solution with the new ProcartaPlex analysis app that features 4PL and 5PL curve fit optimization, provides also group-wise statistical and heat map analysis, as well as export of detailed reports, including publication quality images. So in a recent study, as cited here, both the ProcartaPlex Immuno-oncology checkpoint panel and the human cytokine, chemokine, and growth factor 45-plex ProcartaPlex panel were used to measure baseline levels of in total 59 soluble immunological factors before treatment with a PD-1 inhibitor in plasma samples of a discovery cohort to finally evaluate the response to immunotherapy. So based on a calculated signature score as displayed by the heat map, the correlation between the distribution pattern of these soluble factors and the clinical benefit was retrospectively explored in different subgroups. And as depicted by the scatter plots below, a significantly distinct pattern was identified for these three targets. So while a decreasing trend of therapeutic response was observed for IL-10 across the subgroups, significantly increased pretreatment levels were identified for CCL5, Orantes, and soluble PD-L1. And their relevance as biomarkers for efficient immunotherapy was further confirmed in a validation cohort as depicted in the scatter plots below. And here, moreover, the combination of these two target analytes as so-called two cytokine signature was found to be associated with increased disease control rate and survival, which further underscores the predictive value, which is important for the stratification of individuals who may benefit from a certain treatment. So finally, we continued to expand our portfolio of multiplex ProcartaPlex panel in the cancer research space to ultimately enable the systematic analysis of all relevant targets, addressing all hallmarks of cancer and uncover the complexity as summarized by this rationale and originally published by Hanahan and Weinberg. And besides the readily available multiplex panels for growth factors, for angiogenesis-related targets, and the three immuno-oncology checkpoint marker panels, as just mentioned, that cover all relevant soluble T-cell and natural killer cell checkpoint molecules that are involved in evading immune destruction and tumor-promoting inflammation, and that are now also combined in one immuno-oncology checkpoint marker panel that encompasses all these targets and will be available soon. We further added targets of interest focusing on the hallmark of cancer resisting cell death, where we already provide an apoptotic cell clearance panel and put the particular emphasis on not yet covered hallmarks, which are actually cell proliferation and also metastasis and invasion, that are among the most fundamental features of cancer cells, that enable sustained proliferation and invasive growth capabilities, and are in the focus of research. So now we provide 3 novel multiplex panels with more than 30 highly relevant targets that are underway and coming soon to allow in-depth analysis of these aspects as well. Finally, I'd like to point out and mention the important role of exosomes as key mediators of intercellular communication, particularly in this context, that has gained considerable interest over the past years. So exosomes and their cargo derived from a tumor, but also the tumor microenvironment, are increasingly acknowledged as valuable source of biomarkers and are associated with essentially all of the described hallmarks of cancer and many of the related targets that have been summarized on the previous slide. So basically, to characterize this subset of small vesicles, we have designed and provided a multiplex ProcartaPlex panel, including 6 selected targets as listed in the table, covering all relevant categories of proteins to demonstrate the nature and purity of exosome preparations and extracellular vesicle preparations in line with the recommendations of the International Society for Extracellular Vesicles that is summarized below. So these include actually typical tetraspanins, as well as an additional integrin as transmembrane proteins, synthenin 1, a representative for cytosolic proteins, and cytochrome C as a control. This unique multiplex tool for exosome characterization will also be soon available and will provide an even more complete profile than of cancer immunity and to leverage your immuno-oncology research. With that, we'll end this presentation. I hope I have given you some insights into powerful assays for targeted approaches, but also comprehensive analysis of immune profiles to support immuno-oncology research on the Luminex system. Thank you for your interest and participation in this webinar.